Welcome to the MOOC Interactomics course. Today we will discuss about various parameters that should be considered during image scanning and processing in microarray workflow. Detection probes have been briefly discussed in our previous lectures. These probes are major determinants of findings from any microarray experiment. Detection strategies for quantifying signals from these probes in microarrays have been categorized into two basic categories, label based and label free methods. Due to the strong advancements by DNA microarray technologies in the area of confocal laser scanners, it has been modified and extended extensively in protein microarrays as well. Consequently, label based methods are most widely used protein microarray detection system. Scanners capture the fluorescent label signals using cooled charge coupled device CCD cameras. Some scanners also employ lasers for excitation and a photomultiplier tube PMT detector. In other cases, LED is used for uniform light source and is detected using a cooled CCD in combination. Today we have with us Mr. Pankaj Khanna, who will be talking about GenePix microarray scanner from molecular devices. We will first have a discussion about microarray scanner as well as the software which controls this. We will talk about various concepts as well as live demo of software interface in the subsequent lecture, so that you get better understanding of how this whole process is controlled and can be performed. This demonstration will help you to get a unique learning experience through the user interface of these specialized software. This is my pleasure to introduce Mr. Pankaj Khanna, Manager Application Support from Spinco Biotech Private Limited. Today we will be talking about GenePix microarray scanners, which is from molecular devices, and Spinco is distributor for the same. So we will have a discussion about microarray scanner as well as the software, how to control the hardware. So we will talk about various basic concepts as well as a live demo of the software interface, so that you can get a better feeling about how this whole process is controlled and performed. So Pankaj, welcome to discussion about microarray scanners and the software.
Thank you, Dr. Srivastava. It's our pleasure. At Spinco Biotech, we are having molecular devices as distributors for last many years, and we are dealing with the scanners for many years at the level of microarrays. As you have already described that this is used in the form of microarrays, we are dealing from the image acquisition to the level of data analysis for the microarray applications. So let's go through a little bit on what is GenePix actually can do in a firm. So
Yeah, I think it will be useful if you can just first give some steps for the DNA microarray experiments. All those are proteomics course, and I have been talking more onto the protein microarrays and things. But this whole technology got initiated from the DNA microarrays. So just so that students have some idea about how the DNA microarrays are performed, just give some brief steps about overall DNA microarray technology, and also like how and what GenePix can help in the microarray process.
Sure. So basically the DNA microarray steps are having divided into 6 basic steps, starting from the manufacturing of the microarray. So based on the slide and your biology, the manufacturing of the slide is done. Say if you want to go for the RNA expression chip, you have RNA expression chip made.
Right.
So if you are using a protein expression chip, you are using a protein expression chip. So after the design is being decided, the next step is the experimental design, which usually scientists think based on the biological question what they are asking.
Right.
So based on that, they choose which reference, which controls to be studied and group them. Once you are ready with these two, the next thing comes is the sample collection and the target or the sample preparation, which then can be hybridized to the pre-planned chip. which now is ready to go for the scanning in the form of image scanners.
Right.
So the fourth step involves actually the hardware beginning up, where we as a vendor from the molecular devices come in place, and this data once scanned in the form of raw data now can be made data building, filtering, as well as the normalization, followed by the last steps of bioinformatics and biostatics, which help in the data analysis. So in this fashion, these 6 steps are important for any microarray experiment.
So just to clarify and summarize here, I guess it's the content which is just different, whether it's DNA or protein. But once we have the chips in hand, then entire assay and the whole procedure is very similar. So manufacturing of the microarray will be different for each of the content type. Then experimental design one has to choose depending upon the content and the biological question what they ask. And once these things are in place, then one has to choose what type of biological samples or clinical sample they want to probe, and then followed by perform the hybridization steps. And then once the experiment is performed, then one need to acquire the images, and that's where these hardware and scanners come into the play.
Yes.
Followed by we need to do the data analysis for these, and then the statistical part comes into the play, where we need to identify the meaningful information from these experiments. So these scanners are user friendly and sort of even the cost wise and overall performance wise, they're quite good. They are used worldwide. I had been using it in my research earlier in different places, and also I am continuous user of this scanner. So I'm quite familiar with the overall configuration and the hardware part for GenePix scanners.
Okay. So as you are using 4000B, which is actually the, as you said, the world best the hardware in the form of Psi3 and Psi5 dies being used. Apart from that, people use for different applications as well.
Right. So can we talk a little bit about the platforms which are used for acquisition and analysis of these microarray data?
Sure. So in the form of hardware, as we have discussed, there are a number of different hardware possibilities in the form of GenePix, right from 4000B to 4300, 4400. These are actually doing the hardware part, which allows the lasers to scan the chip. So once we are ready with the chip, so it will be an interface between the CPU and the system, which is allowing the data acquisition to happen. And GenePix is being attached with a software called GenePix Pro software, which helps in understanding how a data is being acquired with the hardware usage. So basically, another software which is also used is Acuity, which is actually a third level statistical analysis software. So in brief, the hardware is being attached with a data acquiring software called GenePix Pro, and this GenePix Pro software is now give the results to Acuity, which can be further analyzed for statistical analysis.
Yes, obviously you need one software which can help in the performing scanning process, and then once the images are required, then you need to obtain some meaningful information from those images, and that's where I think software like Acuity can help over there.
Yes.
So which technology is better for microarray, and what do you think is the more unique feature of using this hardware?
So GenePix microarray as such is based on two special designs. One of them is a non-confocal design of hardware, and second is inverted chemistry.
So what is this non-confocal design?
So non-confocal design is like where you have spots in the same plane where the scanning is done is called as a 2D dimensional. Okay. Say when you go for a tissue arrays or analysis where it is very, very higher, so in the form of spot size, especially at the level of imaging of scanning images, so there at the level of microscopy, people use a technology of confocal microscopy so that they focus the laser at different wavelengths. So at the end of the day, they build a 3D image. But usually for all microarrays, which is the highest is the tissue microarrays, it is shown that all the slides are in the same plane, which is why it is non-confocal chemistry, which just resides in the same plane of the slide. So that is why we choose non-confocal chemistry attached with the inverted chemistry of scanning, which helps in the best results in the form of signal to noise ratio.
So what are some of the advantages of this design of the gene pick stage?
Right. So as we discussed, non-confocal coupled with the inverted scanning, how does it help is usually we see in the glass slides there are a lot of small deformities which our eyes can't see.
Right.
So what we do is we invert the slide so that it goes to the level of the same scanning, and they are supported at the level of the edges with the controllers which see the deformities at the glasses.
Okay.
So what it helps is, it helps in looking at the deformities and directly allow the scanning to happen on the face of the scanning area. So what happens is, because of the confocal design attached with this inverted chemistry, we get best signal to noise ratio so that your data is more valid for any analysis.
So this process can actually resist any misalignment which may happen due to the design of the slide and
Very
overall scanning procedure.
Very true, sir. Very true.
So what are different type of slides which are compatible for these type of experiments?
So as you have already said, these systems are used worldwide, and in different academic institutes, people use at the various academic as well as commercial vendors. So ranging from different applications
Right.
to different vendors, they support, to name few, say for example, Agilent, Nimbolgen, Cornings. These slides are all compatible with them, which usually supplies in the form of either the RNA or the DNA chip. Invitrogen, which supplies the protein chip, is also compatible with GenePix Pro. So apart from lot of Toronto, all chips are also being supported, and incoming tissue arrays which are there, they are all supported with this particular technology as well.
So I think it's very useful to have a platform where one can use actually the slides from different commercial resources. Otherwise, for example, if one wants to use the same scanner for doing DNA arrays as well as protein arrays, many times become difficult. But as you mentioned, like we can also use the proto arrays from Invitrogen as well as DNA arrays from various type of commercial places. So I think it just shows that this platform is quite robust for wide applications.
Yes, it is. Indeed, the latest all applications possible, they are all compatible with GenePix because of this pixelation at 2.5 also, which is now available.
Now, can you just brief some of the applications of the microarrays which people usually perform by using these type of scanners and the software?
Yes. So classically, microarrays are being used for the differential gene expression, which usually people categorize in the gene expression format. So later, as the development has happened, people went for SNP arrays as well as a CGS, which is called as comparative genome hybridizations.
Right.
They are used to look at the chromosomes. Apart from that, protein arrays, chip on chip to look for the control genes, they are also being focused. And the protein arrays are now really catching up in many sense, because the protein is actually the biological functions what many scientists believe,
Right.
and I am also a strong believer. So now people are moving from the DNA to the real functional part, how they are directly affected. The major bottleneck was looking at the antigen antibody reactions, like the proteins are all being seen. There is no general formula, but now the field is catching up. So even at the level of protein arrays, this is compatible. The incoming tissue arrays, which is really in infancy, is also now being compatible with GenePix Pro.
So although like whenever we talk about microarray, people get a feel about it's like probably oligos or DNA based microarray thing which we are talking. But slowly now this overall process from DNA microarrays to protein microarray now field is shifting in that direction. But only major limitation here is that how to generate that content, protein content, in the similar high throughput format what people used to do for the DNA arrays. So major success for DNA arrays was the easy availability of DNA and the stability of DNA, and that's why the high density arrays are possible from the DNA technology. But applying the same at the protein becomes very challenging, because purifying that many protein, keeping them stable and functional, it becomes very challenging. And that's why, like in my previous lecture in the class, I have discussed some of the latest methods which people can use for making protein arrays. And we have talked about different type of technologies, including how one can use the self-free expression based system for generating the protein content, which could be used for the high throughput microarrays for the proteins. So yeah, it's good to hear that there are different type of applications which one can use here, starting from DNA to proteins.
Yeah.
So now these scanners, what type of pixel resolution they can scan, and what is the suitability for scanning?
Right. So our scanners ranges from 5 micron resolution to 2.5 micron resolution. The basic thumb rule for all the resolution is that the amount of your size of your spot, be it protein or RNA, that should be 10 times that of the resolution what you are scanning with. So interestingly, most of the DNA is less than 50 microns, and most of the protein usually stands at 200 microns. So essentially the rule goes that 10 is to 1, if you say 10 is the resolution and 1 is the spot size. So that is a major rule. So just to give you a brief out, say for example, if your spot of protein is 100 microns in size, so what best if you do at 10 microns, basically you're getting 78 pixels. So this is actually 1 by 10. If you do by 5 micron resolution, although the pixelation will increase to 314, but the size of the image actually also increases. So apart from making more than 10 times the size of spot, it doesn't really help at the level of resolution if you increase. The only thing is you're increasing the size, but the data quality is just increased by few very minor changes. So that is why the best rule is 10 is to 1, which 5 to 2.5 micron usually is most suitable, where the people are making the slides.
So there are different scanners which aim for wide variety of scanning pixelation and resolution. But I think what some of the thumb of rule which you have mentioned is very widely applicable for any scanning.
Yes.
So the microarray data could vary in wide range. So how to accommodate that variation while performing the scanning?
Yes, essentially because they are all not present. So the data could be right from the zero intensity, which will be equivalent to the background, to the very highest intensity. The best way to control the variations is looking at a photomultiplier tube. Actually, that is nothing but once you do a fluorescence, so the intensity is coming out of that. So the very low expressing genes or the proteins will give you very low fluorescence intensity, whereas the high will give very high.
Yeah, so just I will interrupt you here. When people scan these microarray slides, it always asks for the PMT gain, right? So I think that's what you're talking here, photomultiplier tube and the PMT gain.
Yes.
So just maybe you can clarify here, like how one can adjust that parameter of PMT gain.
So the basic thumb rule there is in any scanning, you don't want to see a white spot. The white spot essentially means the saturation, and to avoid saturation, exceptional cases for few controls, people avoid that happening. Okay. So the best way is to look at a photomultiplier tube condition. So it ranges actually from something like 300 to 700, which helps one control how much photon multiplication can be done. So the lowest can also be seen and highest doesn't become saturated. So this is how the photomultiplier tube is very essential to control the different kind of variability within the chip which happens due to the biology.
So how to scan a slide for best possible result? You'd obviously like to change various parameters to obtain the best result, right? So what are the things which one need to look for?
Yes, so the major variations actually come owing to the different sources. One could be technical, one could be biological, and another one is owing to the assay or the chemistry. Right. So the technical ones in the form of hardware can be controlled at the level of PMTs, and when you are looking at signal to noise ratio, the small play of PMT and very less with the help of laser power, and then with our own design of inverted scanning, right, as well as the non-confocal chemistry, we are able to achieve the best. So in short, if by visibility you may not be able to look like what is happening to the images. So in the form, there here in the image which is being shown, the which looks brighter to the eye may not be a true sense. Right. So when you look at even the dull, there the background is very low. Without looking at numbers, if I see them, I say this is dull, that is bright. But that is not true always, because when you calculate reality based on what you have controlled at the level of PMTs and others, you quickly come to a conclusion that signal to noise ratio can be seen by eyes at the level of PMT, which the laser balance can be seen. So in this fashion, while you are scanning a slide, you can always look at that.
So one need to definitely adjust the signal to noise ratio to achieve the very good image quality, and obviously simultaneously need to perform the PMT adjustment as well as other parameters.
True. So as you go for the signal corrections, so basically what you're trying to see same thumb rules of not looking saturations,
Right.
and then you expect the image is going to be with least background, so the valid information which is coming in the form of intensities become a true signal.
So how to quantify the signal to noise ratio?
It is very easy to calculate once you have a signal, and then when you have a background which is already calculated. So basically signal is the intensity which is coming from the expected spot, and where there is no spot, that particular intensity which is coming is naive, and there is another way of calculating a local background which is just nearby surrounding area of the spot.
Okay.
That is called as a background. So we calculate signal minus background. So we divide that with overall background deviation, that is standard deviation of the background. That will help calculating a signal to noise ratio.
Now, one parameter which we see for the background correction is basically looking for the scan line averaging. So can you just elaborate on that?
Sure. So basically if you scan once, so image is being collected. So if you want to have multiple scannings being done, so that all intensities can be averaged upon and get to a signal value, it is being seen that when you increase the number of scan, usually people prefer 2 to 3, so then average them. So this helps in reducing the signal to noise ratio in a fashion that you get a real good intensity coming up.
So just maybe you can brief some of the very common features of this scanner, because then only we can learn more when we talk about a software interface.
Sure. So the basic concepts in the form of hardware design, the basic one is non-confocal optics, and another one is the focal depth. The meaning of focal depth is where our laser can focus in the form where it can get or acquire the data, and the depth is 64 microns, which allows a wide variety of applications possible. So if you want to use some slide based slides that is having a cover slips, so that the depth is little bit kind of lower, so you want to focus at a different range. So in this fashion, the non-confocal optics with a focal depth of 64 is our major feature. Apart from that, up down, upside down, that is inverted chemistry, is actually what we are going to use in the form of printed sites being scanned directly and controlled for the variability as discussed earlier, and the resolution at 2.5 or 5 microns based on the system which is available can be also used. And real-time PMT adjustment. PMT we have already elaborated, the photo multiplier tube,
Right.
and the real time is at the level of scanning. We can look at the data when the scanning is live. So in this form we can control the PMT live in between the scan. And then auto PMT adjustment, for those who are not real buffs of looking at how to control them, we have given auto PMT option. This auto PMT option allows Acuity to get the best output, but GenePix Pro allows the multiple scan by itself, so the best PMT suitable for one application can then be seen immediately.
Now, I am a user of 4000B, so I would like to know a little bit more about some of the features for this 4000B, which is more classical scanner.
So GenePix Classic 4000B is called Classic because it is first introduced in 2000.
Right.
But after that, everybody started loving this machine, owing to the fact that it is having 6.5 minutes of simultaneous scan. The meaning of simultaneous scan is both lasers scan at the same time,
Right.
so you have very less time for scanning. Apart from that, it has got two lasers, actually 635 and 532 nanometers, which is classically used for Psi3 and Psi5, and they're on compatible dies.
Right.
So in view of this, we have standard green and standard red emission filters to accommodate all Psi3 and Psi5 applications. The laser power can also be adjusted in the form of 100% to 33% to 10%. So based on what application and what intensity you want best, the laser power can be adjusted as well. So adjusted focal position is from minus 50 to 200 microns. So this allows you to focus in a different waves, so we can have a different slice compatibility coming up with the scanners. 5 micron resolution maximum it allows to go for, and it can go up to 100 microns of resolution. So non-confocal, again, it is using the non-confocal optical design, and any standard slide can be used for the scanning. So it gives a wide applications possible in that.
So data acquisition is always a very important aspect, because once we acquire a good image and good data, then only one can analyze to obtain any meaningful information. So can you elaborate how to acquire the image?
As we discussed in brief, like the GenePix Pro software, we are going to use for data acquisition. So basically, there are different steps involved in the usual process of bioinformatics. The first one comes is imaging.
Right.
So imaging is being done in the form of multi-image or single image TIFF format. So we use TIFF format to create the images form and export these color images in 24-bit JPEG formats. So these JPEG formats allow one to only see, but the basic data processing will be done on TIFF format. So once GenePix Pro is being allowed in a different sense of hardware controls, then image acquisition can be done using the number of lasers availability. It ranges from 2 to 4, and many times few applications use one laser as well.
Right.
So based on the channel type, laser type, you select how many to select for, again on your chemistry basis. And this third-party image alignment and manual automatically being done. So what this means is each particular array has got different blocks, which in turn contains the features. So these features are actual genes or the proteins or the representative of the biological material what you are checking. Now this has to be aligned with the annotation informations. So this can be done by a third party, and the GenePix Pro is doing by itself in the form of automation to do the job. So automated block and feature alignment is possible. Even the different sizes and shapes of the spot can be handled, right from circular, square to irregular features, which can be handled at the level of image alignments.
So I think you have mentioned the good points about data acquisition, because one need to look at various type of features and different type of analysis later on which one need to perform. So the data acquisition has to be very flexible in terms of the sometimes spots could be irregular and there could be different type of background, there could be different type of chemistries being used. So I think by looking at some of these parameters, one can acquire a good image, and then the data acquisition can be further used for further analysis. Yeah, so maybe we can talk a little bit on the analysis aspect.
Sure. So in any microarray, after the image, first thing comes is the alignment, which we have just covered in the form of data acquisition, because that is a very important part to cover. And apart from that, you have a background subtraction so that you get a true signal coming in. So GenePix Pro does help in the background subtractions in a different format, and also in the normalization of the features. So if you see, the background subtraction can be done in the form of local, global, and negative or morphological control. So these negative and morphological controls are subjected to the design of the slide type. Say negative controls in your slide, there should be some spots which should not bind to anything and leave blank. And whereas the local and global can be calculated in the general space where there are no spots available, and the area which is not being spotted nearby your particular feature. So in this fashion, background subtraction can happen, and then feature viewer and feature pixel plots. So basically the major thing comes in the acquisition is, after acquisition is visualization of the data, and this visualization comes in the form of pixels and the plots, the graphs. So graphs help us in understanding globally what is happening in short, so it gives you a real image how the things are happening. So this can also be done, and there are multiple ways of calculating in the form of ratio calculations after normalization or during the normalization of the data. So analysis immediately after those involve few of the normalizations process, which GenePix Pro can very well handle. And other important feature is the flagging of the spot. in a biology we see some spots are really not good, or because of some artifact they are not supposed to be taken for analysis.
Yeah.
So essentially we need to make that spot as flagged as good, bad or absent. So these can also be done with GenePix Pro software. Lastly, normalization of the images and the different formats is also allowed to happen in GenePix Pro.
I think you rightly mentioned the need and the importance of the control features, because many times like whole experiment is the quality of the experiment depends on how well your controls have performed. That applies to both negative and the positive control. And when you talk about DNA microarray technology, obviously we are talking about very high density arrays here, where a lot of spots and a lot of controls are already inbuilt in place, right? Many times when I talk about protein microarrays, and especially when we talk about functional type of arrays, so we actually put different type of controls just based on that one particular experiment. For example, I'm looking for some biomarker response. I need to have certain positive controls, some biomarker which need to light up on the array, which will guide me as a positive control. Then if I'm looking at immune response, I need to have some sort of IgG and some of those type of control features which will guide me to just how non-specific the response could be. So again, some empty spots we need, some type of spots where there is no DNA or no protein is printed there. So no biological material is there. So a lot of different type of control features are usually part of these array design, and that helps further for background subtraction as well as looking at how good quality data we are obtaining.
True. So that is a major calculation for the background one.
Right. So let's talk about visualization, like how one can really generate some good image and which can be further used for analysis.
Right. So once the image is being acquired, so immediately scientist wants how my data has performed. As you are given a
Right.
very beautiful talk on how the controls works, and especially few people use even the bacterial controls, which is non-related to the biology as such. Totally they want to see just the assay controls.
Mhm.
So first they say whether my assay has worked or not. So
Right.
best way to do for them is looking at the graphs which are being given differentiation in the level of backgrounds, and though also can be done at the level of different histograms and the scatter plots possible.
Right.
And these scatter plots can be plotted once against the channel type, say laser 1 versus laser 2 or wavelength versus wavelength 2, in classical say Psi3 versus Psi5. So how these two things have behaved for me.
Right.
So again different kind of graphs, and so also the images can also be exported to PDF as well as being visualized in GenePix Pro for your further screening for different QC applications.
Thank you for this interesting discussion on the basics of scanning parameters and image generation. I think our students are now stimulated for our next lecture, which would have the live demo of the GUI surrounding the specialized software used for image scanning, processing and analysis. Thank you.